So we're going to be talking about some of the advances that have been made and are being made in labs here at Northwestern as well as at the University of Chicago and beyond, based on tissue that comes from patients like Susan and cell lines, animal models, et cetera. So we're going to hear from Katie McCortney, who is here at the Biobank at Northwestern, and who is involved in actually procuring that tissue so that it can be used for research. And then we'll hear from Dr. Deric Park, who is a longtime chordoma researcher, and he's going to tell you about some of the advances coming out of his lab, as well as a survey of some of the exciting science happening in other labs across the world. So then we'll have an opportunity for a Q&A after that, and I ran over my time allotment, so didn't have a chance for Q&A after my talk, but we'll be happy to take questions after these two speakers as well. So without further ado, Katie.
Hello, everyone. I'm going to apologize up front. I got a head cold about 2 days ago, so if I sound a little hoarse and gravelly, apologies. But as Josh mentioned, I'm here to talk to you about biobanking and the importance related to research as a whole, but in particular for chordomas. We're also going to talk a little bit about the molecular underpinnings of chordomas, so I'm going to bring you down into the weeds of the science world and try to help you make sense of some of the things that we've heard about throughout the day today. Did I do it wrong? There we go. Okay. So first, I just want to talk about what is a biobank. A biobank's primary goal is to supply researchers with high-quality biospecimens. And when we think of a biospecimen, that can be anything removed from the human body. So this can be anything from a blood sample to your tumor tissue removed during a surgical procedure. Human biobanks in particular allow researchers access to valuable tissues, as we've been hearing a lot about today, so that they can be used in their experiments. Our biobank is a little bit unique compared to some others across the country and across the world, in that we collect our tissues directly from the operating room. So we work very closely with Dr. Walensky and the rest of the attending neurosurgeons and the rest of the operating room team. So when your tumor is removed, it's always to the discretion of the surgeon, and any tissue that they deem in excess from diagnosing your tumor appropriately, that's then allowed for research purposes. Of course, all patients are consented prior to their donation and prior to surgery, and a single patient donation can affect several different research projects. Okay, so our biobank here at Northwestern is called the Nervous System Tumor Bank. So we cover all central nervous system tumors, so everything in the brain and everything in the spinal cord. We've collected approximately 1,850 tumors and matched blood samples since the beginning of 2014. You can see our graph there. We've had quite a positive increase over the years. We support over 80 active completed research projects from about 30 to 40 investigators. And those investigators are both here within Northwestern and other outside academic institutions. Of these, almost, as of this month, we're closing in on about 2,000 tumor samples that we've collected. Only 14 of them are chordomas. And of those 14, 10 are spinal, and I think all 10 of them, I believe, have come from Dr. Walensky's surgeries since he joined the Northwestern team. And 4 of them are clival, being of the base of the skull. So the director of our biobank is a pathologist, so we're going to talk a little bit about the histology of chordomas and what those look like under the microscope. On the left, you'll see a classic chordoma, which is described as having bland-looking cells in a myxoid matrix. The myxoid matrix is going to be that light blue background you see on the slide there. The cells are arranged in clusters and cords, chordoma. And in particular, a lot of the cells look bubbly. So bubble bearing is a very common feature to chordomas. On the right, this tumor is similar, but the matrix is a little denser and is a little more cartilaginous. This is then a cartilaginous chordoma. These features, along with a cytokeratin stain, are important to distinguish between higher and lower grade chordomas as well as lower grade chondrosarcomas. And the one on the right there, you can see the darker blue hue in the background there. That's because the myxoid matrix is a little bit more dense than the one on the right. So now I'm going to bring you down into the weeds of science, and we're going to talk a little bit more about the molecular underpinnings of chordomas. So this paper is back from 2010 and was authored by the director of our biobank, Dr. Craig Horbinski. And this was published back when he was in Pittsburgh, where at the time they were well known in the chordoma community. Approximately 10% of the chordomas in the country were seen there for to be surgically addressed. They had access to 28 clival chordomas, again, clival being of the base of the skull, and those were accessible to them through a biobank, and they found two significant molecular markers for these chordomas. First, they found that a low Ki-67 proliferation index was favorable, and this makes sense because the slower the growth of the tumor, the better off we're going to be. Second, they found that an intact CDKN2A gene had the same association with better overall survival. The CDKN2A gene is located at chromosome 9p21, and we're going to circle back around to that, so keep that in your brain, but I promise there's no quiz, so no worries. But what does this all really mean? We have these Ki-67s, we have CDKN2A. What do we do with this information? We're going to talk a little bit about basic biology. We're going to talk about the cell cycle for just 1 minute. The CDKN2A gene encodes the P16 protein, which you can see in the blue bubble there. And P16's job is to block cyclin-dependent kinases, which I'll refer to as CDKs. And this prevents the cell from entering the cell cycle. So when your cells are dividing, they have to enter the cell cycle to duplicate their DNA, create their sister cells, and so on and so forth, and create multiple copies of each other. When P16 is deleted, so there is no regulatory body telling those cells, don't divide yet, that's when your cancer cells can divide as freely as they want, and your tumor can really kind of ramp up its activity. So this next paper built upon the previous paper from 2010, but you can see here this reference is already from 2019. So we've already jumped forward now 9 years in the literature and discoveries related to chordomas. They explored further molecular panels for chordomas, and again, this data was generated through the use of specimens available through biobanks. The previous paper noted the advantage of the intact CDKN2A, which, if we recall, was the 9p21, which encodes that P16 protein. And this paper adds the additional finding of the benefit of also having an intact 1p36. And that's sort of summarized in the box all the way to the right. But again, what does this mean? These are numbers, these are letters. What do we do with this information? The truth of the matter is, as Josh had mentioned earlier today, we're still figuring this out. We're still learning. The molecular markers for other tumors and other cancers have been far more explored and are better understood. And at this point, no one quite knows what that 1p36 gene is doing within the tumor. But again, with available access to these biospecimens, researchers will hopefully find the reason or the cause and maybe be able to target that in the future. This next paper worked with chordoma cell lines, which we just heard a little bit about. And that's also something a biobank can work with researchers to help establish. Here again, we see the importance of the P16 deletion. So that means your cancer cells are ramped up and they're dividing really quickly without any regulation. And the cells are more likely to go into their cell cycle. The good news, though, is that these tumors are more eligible to be treated with palbociclib. Within these cell lines, the researchers found the more palbociclib administered, the less the tumors grew. So palbociclib works by blocking part of the cell cycle as a CDK4 and 6 inhibitor. So those CDKs are those, now my head cold is messing with my memory, cyclin-dependent kinases. that are also involved in the cell cycle. So the palbociclib essentially creates the same effect as having an intact P16 gene. And palbociclib is used to treat a lot of other cancers. Its use on P16 deleted cancers is expanding. I think, if I remember correctly, it's most widely talked about and commonly used in breast cancers that are HER2 positive. So this drug has been out on the market for many, many years. And toxicities and things like that are well understood. So as Josh was talking about earlier this afternoon, this use of drugs that have already been produced is a really excellent avenue for furthering chordoma treatment. In fact, there's a clinical trial in Germany using palbociclib on chordomas. This group is in Heidelberg, Germany, and they're currently in phase 2. Okay, so another more recent study utilized a mouse model using cells from the Chordoma Foundation and Massachusetts General Hospital, both of which have their own biobanks. They found that all chordomas in their hands needed brachyury, which is also something we've heard a lot about today. And brachyury is a protein that gets into the nucleus and turns on transcriptions of specific genes. Specifically, it turns on several of those cyclin-dependent kinases, or the CDKs, which we've been talking about. They then used THC1, which is another type of CDK inhibitor, although it acts differently than palbociclib. So both THC1 and palbociclib are the same type of drug, but they have different functionalities. And they also observed that THC1 slowed down tumor growth in mice, so potentially another treatment option for chordomas once it's been further tested and further examined within the laboratory, and then potentially moving into a clinical trial. This paper here is authored by Deric Park, who will be speaking immediately after me, and they studied, again using cell cultures, that they established from 20 out of 23 chordomas. And I'll just pause for a minute to note how extremely successful that is within a lab to establish cell cultures in the majority of your tumor specimens. Establishing a cell line can be very difficult. Patient tumors are never the same. You are all very different individuals, and all of your tumors are very different. So for this lab to generate such a high percentage of cell lines is really something that's incredible, and a lab that should be very proud of what they did. Anywho, they identified a P10 loss in 25% of the cell cultures, which is also a tumor suppressor gene, so something we want the tumor to have, which matched up with a high proliferation index and then a poorer outcome. They also found that targeted inhibitors using PDGFRA inhibitors and HDAC inhibitors in combination, so using those two together, were more effective than either in isolation in slowing down the tumor cell growth. So again, what does this mean? I just threw a bunch of letters at you. What do we do with this information? These types of studies, finding that drugs in combination working better together, this is what's going to drive the research in the future, and this is what will drive treatment options for new patients and current patients and recurrent patients. So overall, all of these papers needed biobanks to supply them with those biospecimens, and all those biospecimens came from patients like you that consented and signed their name on a sheet of paper and said, yes, you may use my tissue for research. With the biospecimens, researchers and clinician researchers are able to explore the molecular underpinnings of chordomas. Understanding these molecular features is a continuing effort, and as I've mentioned, as Josh has mentioned, the desired goal is new therapies, because as you all know, that's something that's not currently available. Here at Northwestern, our effort is always continuing, especially with Dr. Walensky and his team, and the rest of the neurological surgery team. So we'll keep pushing. Researchers love to find new solutions for things, and we get really excited when we do. But having the access to those specimens, that's really paramount in driving some of these things forward. Here are our lab members. We have 2 full-time staff members within the NSTB, the Nervous System Tumor Bank, myself and our lab technician, Rodrigo Javier. And then a thank you to all of our funding sources and a thank you to Dr. Walensky and the Chordoma Foundation for putting on this conference today. Thank you.
Katie, thank you for that wonderful talk. So, up next, Dr. Park, whose work you've heard about a little bit. Dr. Park is going to give you a survey of some of the latest advances in chordoma research, both in the lab and in the clinic.
Thank you, Josh, for inviting me, and also JP. So I'm from University of Chicago, a crosstown competitor, rival, but no really collaborator. I started work in chordomas. I'm a neuro-oncologist and clinical training, laboratory training. I started working chordoma back in 2007. Josh came to me along with his mom because we took care of him back in Pittsburgh. And you guys know Paul Gardner, some shoulder surgeon. So Paul was one of our residents at the time, and Craig Korbinski was also one of our residents in pathology at that time. So Josh and Simone, his mother, came to me and came to my research lab and said, you guys are really doing interesting stuff with glioblastoma. We had interest in hypoxia, low oxygen. Like, why don't you work on chordoma? I said, why the hell do I do that? First of all, there's no model, right? We had one cell line. The second cell line turned out to be rodent. It wasn't even a human cell line. So we don't know anything about it. It's too rare, but somehow they convinced me. And we Oh, okay. Does it work better? Yeah. Thank you. Okay. So I'm going to talk about a couple of vaccine platforms that are available. These vaccines both target brachyury. And so we all know what brachyury is now and how important it is. And the reason it's so difficult to target brachyury is what we call transcription factor. It's a protein that regulates, turns on and off DNA. So it's not a traditional protein where it binds to another protein. You can inhibit that. It's not an enzyme, so it's very hard to target brachyury. So the strategy here is to use a vaccine to downregulate brachyury. Okay, and there are two different platforms. There's actually more than this. And you may wonder why companies are interested in brachyury because chordoma is so rare. It's not because of chordomas. It's because certain cancers, when they invade, they turn on brachyury to allow them to metastasize. So, but chordoma is a great model to study because all patients with chordoma express brachyury. If we don't see a signal of chordoma, we're not going to see it in breast cancer or colon cancer. And I'm going to also get to our strategy to try to enhance radiation. We know that radiation is very effective for chordomas. And then some medical therapies. I'll skip this. You guys all know what chordoma is, what it looks like. I'll skip this too. Okay, so I'm going to start with the TRICOM, MVA TRICOM. The reason that we target it is because University expressed. There are two different platforms. One is using virus to deliver brachyury. Another is using a recombinant yeast. Recombinant just means recombining. So we're taking yeast and adding the human gene into the yeast to grow it and to make brachyury. And he killed. Okay. And the idea is both of these studies combine radiation with a vaccine. The idea is that when we radiate chordomas by killing the cells or causing what we call immunogenic modulation, it allows the tumor to become more immunogenic. The immune system will recognize it more readily. So here's a cartoon that I borrowed from my friend Jim Hodge, who actually developed both of these vaccine platforms at the NCI. The idea is that we radiate the cells, some cells die, that allows the tumor antigens to be upregulated. The immune system now can go and home in and target and kill the tumor cells. Okay. Similar cartoon, I'll skip. I'll go to the studies. Okay. So this is the work that Jim did in his lab in showing that on the very left, you see chordoma cells, chordoma tissue, and the dark areas are staining for CD8 T cells. So these are the killer cells that we want to upregulate to induce damage to the tissue. You notice that with no treatment, there are very few immune cells in the tumor. If we get vaccine, it increases the number of cells that migrate into the tumor goes up. Radiation alone has a modest effect. However, if you combine both, you can see that there's a robust increase in immune cells, and this is the rationale. Okay. So the first study is called TRICOM, and the reason it's called TRICOM is it stands for Triad of Co-Stimulatory Molecules. Co-Stimulatory molecules are molecules that make immune cells more active. So we want to increase inflammation. Earlier today, somebody had mentioned, I think it was cannabinoid that has an anti-inflammatory effect. Well, in this instance, we don't want that. We want to increase inflammation. What nivolumab, pembrolizumab does is a very non-specific way to upregulate the immune system. You're taking away the brake. So the immune system will attack everything, including yourself, which we want. Right? And we use two different viruses for this. One is called Fowlpox, and then another one is called modified vaccinia virus. They do different. The reason we combine the two is that your body will become tolerant eventually. So by combining two different vaccines, we try to stimulate the immune system even more so. Yeah. So these are the three different TRICOM molecules: LFA1, ICAM, P7.1. So that's the yeast. That's the viral. So we just had really good news. I could share this news, right? So study was done looking at. This was a phase two study. Majority of patients were enrolled at Mass General, and we had one patient that showed dramatic response 3 months after radiation. So it could still be radiation effect, but however, 3 months is a little early. We typically don't see tumor shrinkage until at least 6 months or 12 months. So this study now it'd be reopened, but I'm not exactly how many centers. We will be opening this at University of Chicago very soon, and also be available at Mayo Clinic, both Mayo Clinics, Mass General, and variety of centers. I'm sure the Chordoma Foundation will send an email to everybody. The second approach is using what we call tarmogen. So this is the yeast. So we take the human brachyury gene, put it in the yeast, we grow it up, and then we take the yeast and we kill it using heat, but the protein is still intact. Then we inject this. This is a study that we ran at the NCI. Currently, the study is closed. When I left NCI to come to Chicago, I just met with the president of the company, and they are very eager to reopen this. So hopefully, we'll engage the study soon. This is a study. So this study is randomized, and it's placebo-controlled, double-blinded. So I don't know what patients are getting. The patients don't know what they're getting. The other study is open. It's not randomized. There's no placebo involved. Okay. Here's an example of a patient, Clival patient, for this brachyury, the yeast study. You can see on the left, this Clival tumor. And then over a period of time, it shrunk dramatically, which is quite rare for radiation alone to do this. But again, this patient is still blinded, so we don't know what this patient has. Here's another one. So this one's interesting. So this particular patient was on the study. It was actually coming from Amsterdam. Would travel every other week, flying in and out to the NCI. And the tumor started growing. So at that point, we broke the blind, and the patient was on placebo. And then, then he had an option to cross over now to get the vaccine. So we started giving the vaccine, and he emailed me after a second visit, so 2 weeks after, he emailed me said his pain started going away. And he said, I came off my opiates, and then by 4 weeks, he came off all medications. And then we saw this response, which is quite encouraging. Okay. So I'm going to briefly touch on our efforts to enhance radiation, to make radiation more effective for chordoma. This is a drug called LB100 that it was developed based on work that we did many, many years ago when I was a postdoc at the NIH. What this drug does, it seems, is when cells are growing, when they divide, they go through what we call cell cycle, but you heard that briefly. What this drug would do, it'll make the cell cycle arrest at certain stages, what we call G2M arrest. At this particular state, the cells are much more sensitive to radiation. So we started combining with the radiation. Here's a graph here. You can see that on the very the black one is a control, so we're not doing anything. So everything is normalized to this particular population. When you get the drug alone, it does hardly anything. And then when we get radiation, there's a modest effect in cell growth. However, if you combine it, you can see that the dramatic decrease in cells, at least 50% within 72 hours. This is a slide. I'm going to try to avoid jargon, but if you guys have any questions, please don't hesitate to raise your hand. So this is a stain that we're looking at. This gamma H2AX, all it means is a marker of DNA damage. When it's called double-strand break. So what radiation does is using oxygen, by giving radiation, oxygen becomes toxic, and that leads to DNA damage. This is what we're seeing here. And then you can see the blue, it's really hard to see the green dots, but you can see that green dot, that's the marker of double-strand breaks. It goes up when we combine this particular drug along with radiation. Okay. And this is a graph showing, it's from animal study. Black is just nothing, okay? And then red is radiation alone. Then you can see that if we combine radiation along with this drug, and depending on RT dose, there's dramatic decrease, attenuation in tumor growth. So these are rodents that have no immune system, because if we use an animal with immune system, we try to implant human tumors, their own immune system will get rid of it, will reject it. So we had to use mice that lack immune system. And I think this is another reason it's so important what Josh is developing what's called PDX models. So these are tumors that are removed from patients like yourself and directly implanted into mice. Unfortunately, this study, we use this particular cell, like a UCH1. This cell has been passaged in plastic many, many times. And we know that when we grow cells, they change over a period of time. So it's not a perfect model. And this is the size of tumors. You can see that. Again, green is combination of radiation and this drug. Blue is a higher dose of radiation. So there's a definite dose response. And these are the tumors that we actually go and take out, harvest. And you can see the size. So vehicle, again, is control. LB100 is a drug alone, radiation, and then you can see the combination tumors are much, much smaller. So this paper was published about a year ago, I believe, and Dr. Gandhi had met this morning. He runs a proton center, so he's very interested in proposing this study and translating this to the clinic. And I think with the 10 proton centers he had discussed, we could quickly accrue patients and answer this question. And another attractive thing is that this drug has already gone through a phase 1 trial in human, so it will not be a first in human study, which will facilitate the clinical trial. Then we talk about, I was going to discuss brachyuria, but since Josh already went over that, I'll skip that part. This is a study that is in press right now. We just published. It should be coming out within this next month or so. So we're asking the question, why are chordoma cells so resistant to chemotherapy? Because all chemotherapeutic agents basically fail. It doesn't work. Even toxic combinations that we use for sarcomas, using platinum or using Adriamycin, it just doesn't work in chordomas, unfortunately. And so we thought there must be a way that chordoma is protecting itself from toxic chemotherapy. So we did what's called an unbiased study. So we gave chemotherapy and then used a library of thousands and thousands of compounds to see if there's anything that may make the chemotherapeutic agent more effective than chordomas. And what we identified was were several hits. Josh mentioned hits. And these are all pathways that protect the cells from DNA damage, called PARP pathway. So we thought, oh, this is interesting because this drug is available, olaparib, and there's newer generation drugs. These are often used for breast cancer. So again, if there's a drug that's already available, it's much easier to take it to clinic, so we started looking into this. So we found this panel shows that chordomas don't respond to chemotherapy. These are even chordoma cells that we grow on plastic, they don't respond to chemotherapy. And you can see in panel D, TMZ is a chemotherapeutic agent that we use for brain tumors. You can see that UCH1 and UMCOR1, those are both chordoma cell lines. With chemotherapy, we don't see cell death. Apoptosis just means cell death. They're about the same. It doesn't do anything. You can notice the U251, U87, these are brain tumor cells. They respond to chemotherapy. So why is it the chordomas do not? Again, so this is showing our screening pathway. And the hits that we see on the panel B are the DNA protective pathway. And then when we combine the two, let's see here. So before we went to drugs, so if you look at D, this is what's called a comet acid, which is looking, it's a smear. That means DNA is being damaged and being spread across. And SI just means, I think Josh mentioned earlier, is a way to block protein expression by blocking the RNA. So DNA is a blueprint, right? And protein is the structure. So if you're building a house, you need an architect, you have to have a blueprint, that's your DNA, and then the actual house is the protein. But between that, there has to be a message. How does the blueprint instruct the person to build the house? That's your RNA. So by blocking the message, we could block the protein expression. So here at the very bottom E, we looked at 3 different cell lines. If we use temozolomide, it just means negative means nothing, positive means we use that. Temozolomide alone, which is the 4th one, does very little. However, if we combine and we use 2 different ways to knock down the gene, and you notice that if you combine temozolomide with a PARP, either 2 different molecules, there's dramatic increase in this tail of DNA, telling us that there's now DNA damage. So because the drug is available, so OLA means olaparib, so it's actually a drug that targets this DNA repair pathway. And you can see that if we combine it in B, there's dramatic response. Cells are no longer viable, they die if we combine both the chemotherapeutic agent and this particular drug. And you can see it in D that, if you look at D, the second blank column is drug alone. It causes very little cell death. And then if we just use just olaparib alone, that's the 3rd one. Again, very little. However, if we combine both in all 3 cell lines, the dramatic cell death, up to 90%. And of course, we had to validate this in animals. We, again, grew chordoma cells, took mice that lack an immune system, implanted tumors, and because chordomas grow so slow, we have to often wait for almost a month before we start treatment, until we actually feel the tumor, or we put load of mice in MRI machines. We can actually measure the tumor growth. Yes, I saw somebody laughing, but it's kind of a sad state. You know, if you're a mouse, we get an MRI. So some of our patients can't even get MRIs, right? Pardon me? Oh, we anesthetize. Yeah, we just knock them. Yeah, we have a whole system anesthesia down there to do that. Yeah. So you can see here, this is a graph showing tumor size. Now we're measuring the directly tumor size. Again, DMSO just means controls nothing. We're just giving basically salt water, not quite, but similar to that. And the OLA is olaparib alone. It's very similar to doing nothing by itself. It doesn't hardly do anything. If we give temozolomide, there's a modest decrease in tumor size, slight. However, if we combine it, we virtually suspend growth. That's the blue. And then D is showing tumor size. So I'm going to end here, and I'm happy to take questions. To date, as you know, only effective therapy of chordoma is surgery and radiation. And that's why this morning my talk was so brief that there is no state-of-the-art medical therapy, because nothing has worked thus far. However, and I think this is really critical, especially being such a rare tumor, that nobody could study this on their own. We really need a collaborative team effort. And I think Foundation has done an incredible job bringing us together that allowed this to happen. We are very hopeful within the next several years we're going to have therapy available, and our goal is really to make chordoma a chronic disease. So thank you.
So I'll invite Katie up for the Q&A. Does anyone want to lead off with a question? Are there any questions? Richard. Yeah.
I've asked this question before, but I've forgotten the total. Am I on? Yes. So, frozen cells from 7 years ago, are they useful, and what can they be used for? They're at Mayo Clinic from a biopsy, the original needle biopsy. Yes.
Thank you. Most cells, if they're frozen with best practices, which are widely available to biobankers, then yes, they're still viable. If they're thawed properly and treated properly, they'll come back and they'll grow. They can grow on a plate. They could grow as a cell line. It depends a little bit on what preservative that they're in, but for the most part, yes, they're still viable.
And so how does one go about doing that then? Or
Growing
how do I
them?
instigate that?
Do you want them?
Yes, and you have them.
Sorry,
How does he get his cells from Mayo?
how do you get them back in your own hands?
To get it somewhere else?
Good question.
The Chordoma Foundation.
Yeah, we should talk. So through our biobank, we have the ability to consent patients anywhere in the country. A challenge that individual centers face is that typically their biobanking protocol only applies to their particular center. But if you have samples stored, we would be delighted to have them used for research, and we can definitely follow up. Patty would be the person to follow up.
It would depend on how it was frozen, because typically, unless they're studying the disease actively, they want to just freeze a chunk of tissue. In that case, it's not viable. It's still useful. We could use that to study, but you can't grow the cells from that. Because as soon as you freeze cells, you basically fracture the membrane. When we freeze cells to use for research, it uses like she mentioned, very particular preservative that protects the cell membrane to prevent it from breaking apart.
Okay.
Because the water molecule in there will freeze and crack everything. So we have to use a very specific way to freeze it. And you can't do that with tissue. It has to be individual cells.
I'm going to add a little bit to the complexity of this problem, right? I don't know if it's a problem, but there's a complexity of getting the tissue. And that is that, when we take a specimen out in the operating room, it's different for the clival chordomas because we're going intralesionally with them. So we can save some of that for biobanking almost every time. But if we're doing an en bloc resection, it's also a coordination of the pathologist and us making sure that we can verify the margins before we take a sample, right? Because we don't want to take away the information that we can provide you as patients by contaminating that tissue before we take it out. So it's a balance. And so like when you look at the slide of the chordomas that we've collected here, there were 10 in that slide, but there have been more that we've operated on. And so some that are within the bone that we just can't get to without completely destroying that specimen before the pathologist is able to render the diagnosis for you. We just feel ethically we can't really do that, right? And so it's a balance, right, of how we do that. And then the other complexity comes through of how do we get the Chordoma Foundation tissue to them and satisfy all these other banks, and that's a balance too that's always like a headache at every different institution of how we get this tissue out to the Chordoma Foundation.
I'll just add one more thing too. I think I think I'm understanding your question a little bit better. So if your tissue is sitting, and this goes for anyone with tissue sitting anywhere in the country, they're called material transfer agreements. And so the guidelines that each institution has to follow in terms of sharing, whether it's data or tissue samples, varies from institution to institution. So you could hypothetically call up and say, you know, I donated tissue here 7 years ago, and I would like that tissue to now go to the Chordoma Foundation. And depending on what their regulations are, they may be able to initiate paperwork to make that happen. But again, that varies institution to institution.
I think Sharon has one, but make sure you hold the mic close to your mouth because people on live stream can't hear us.
And then Mark. One thing all this tissue talk is making me think about is profiling tumors, which was something we talked about a lot at the past conferences. And even with 18 surgeries, I never had any tumor profiled because it was too early. But we haven't talked about that at all. So is that not as important anymore because we've learned new things?
Well, I think this is a question for Deric in particular.
What was it? Go ahead.
About tumor profiling. Where are we in tumor profiling? Is it recommended for chordoma? I don't know if you want to take that.
This is something that we struggle often for because majority of my patients are glioblastoma patients. You know, a lot of foundation and Caris, these companies have a lot of money doing this. I don't routinely recommend it because if you actually look at the Foundation Medicine report, it'll mention all the mutation and it'll say targetable, and list these drugs, which are absolutely ridiculous. These are drugs that have already failed. Just because it happened to work in that pathway in plastic doesn't mean it's gonna work on you. And not only that, often my patients are stuck with a huge bill. And I was talking to Josh about this, is there are certain markers that seem to may help predict how aggressive the chordoma is going to be. I think those are helpful. That could be done at most any centers that do chordoma surgeries are able to do these studies. And trying to get additional targetable hits using foundation, I would not recommend that. It's not worth it.
So it's a very good question, and just to kind of build on what Dr. Park was saying, I think probably many are aware that there is a great deal of hope and promise that is talked about with respect to tumor profiling. And I think when it first came out, that the hope and promise was even higher. People thought that this was going to be the answer, that all of a sudden you have this explosion in targeted therapies. Now you can read out the alterations in the tumor. Let's match those targeted therapies to the tumor. And I think two things turned out to not be favorable for that to occur. So one, in the case of chordoma has a quiet genome. In other words, there aren't that many alterations in chordoma. So even the number of patients who have these profiling tests done that show up with an actionable mutation is relatively small. It's not to say that it doesn't happen, but it's small. So Dr. Gounder at Sloan Kettering has studied this, and I believe he actually has a paper coming out on it. And so I think just to be complete and fair, there are definitely cases where they have found something, and that something, that actionable alteration, has led to a beneficial outcome. But it's definitely the small minority. It's not something that is necessarily going to work for everyone. But then I think also, as Deric was saying, these actionable mutations, you can't necessarily draw a line from actionable in one disease to actionable in another disease. So just because it was actionable in, let's say, colon cancer or breast cancer doesn't mean it's going to be actionable in chordoma. I remember asking this question of one of our medical advisory board members, and they shared the story of there's a type of inhibitor called a BRAF inhibitor, which works in melanoma but doesn't work in colon cancer. Same gene altered, works in one disease, but doesn't work in the other disease. And I think there's been research to show why that's the case. But so you can't necessarily draw a straight line between actionable alteration in one cancer and activity in chordoma. Yeah, Dusty.
I just have a comment. Most of the clinical trials seem to want you to have a genetic profile from Foundation 1 in order to get into the trial. So whether it shows anything or not, it seems like, well, you can't get in unless you give us this profile.
So there's a little bit of a catch-22.
Yeah.
I think there are some trials that are designed specifically to answer the question, does an alteration in a particular gene matched with a therapy that targets that alteration, does it actually lead to benefit? And so I think just to be very, very clear, our medical advisory board has kind of come down with a very nuanced recommendation, which is that as a general routine, we can't recommend at this point that everyone have tumor profiling, but if you've exhausted other treatment options and you have the opportunity to participate in a clinical trial for which tumor profiling is included, then that's a really good thing because we'll learn something. We'll learn whether it's going to work or not.
But also, clinical trials will not mandate foundation. And if you have the type of tumor that the trial is interested in investigating, that trial center will take your tissue and test it themselves. They will never mandate third-party profiling. They'll never do that. First of all, it's not effective, and secondly, it's not ethical.
The other thing about this stuff is it had been in the past very expensive to get these
Tumors profiled, but it depends where you are regionally. We happen to be very lucky here in Chicago, luckily, or however you look at things, but I think it's Tempus and Foundation Medicine that they're doing these profiling, and one of them, I can't remember which, is owned by the guy who does Groupon, which is based in Chicago, and he has a relationship with our Cancer Institute. So for our patients, we requested I think it's very cheap. I mean, relative to...
I don't know what shows up on insurance, so I'm not sure.
Yeah.
I think we have a question back there.
Yeah, my question was, you talk about sharing these tumor samples. You know, in my case, I donated my tumor to Mass General in Boston. How big of a sample are you sharing, and how many places does that go to how many times can you cut that tumor up and send it around?
Good question. So it depends on a lot of factors. So it depends on how big your tumor was to begin with. And it also depends on how much the surgeon decided to give to us researchers. So if we establish a cell line from it, that cell line, if it becomes able to reproduce and grow indefinitely, it could go everywhere. You could be very similar where your cells are all over the country. So it depends on the size of the tumor. We have some tumors that go out to 20 researchers. Other tumors only go out to 1 or 2, depending on the size. For cell lines, I have a whole biobank of cell lines frozen down that somebody could come in and request those, and those could go anywhere in the world as well. So it's difficult to estimate, but there is always the likelihood that your cells are somewhere else.
I think we have
Doing good.
I think we have time for one more question. Yes.
Thank you. So, my name is Chris, and I just want to follow up a little bit on the donation thing with the foundation. My personal experience was to donate some of my husband's tissue a few years after his surgery that had been at our local university hospital. On the foundation site, there's a part about the biobank. Patty Cogswell is the contact. She provided me with a paper that was basically a release that I had to fill in some information. Then she was able to follow through and get the samples. So it's very simple for any of you to make a donation, and it doesn't have to be a fresh surgery today. That's something that is kept, my understanding is for years, where you may have had your surgery. And I'm sure Patty would be willing to help you make that request and get that donation to the Foundation Biobank.
Chris, thank you so much for mentioning that. It's absolutely right. And different ways in which the tumor is preserved enables it to be used for different types of research. So if it's paraffin embedded, there's a certain number of experiments that can be done with it. If it's frozen, there's other experiments that can be done, but it's all useful. So yes, if you go on the website, there's information about the biobank. You can contact us just at support@chordoma.org if you have any questions. But also Joan, our Director of Research, is here. So if anyone is interested, please see Joan during the reception. She can tell you more. So a round of applause for Deric and Katie.